I will start in a couple of minutes. Right, so I'll share my slides. As always, the session will be recorded and uploaded. Right, so this is the sixth live session for the course NMR spectroscopy for structural biology. Last week, we had just started to go into the different structural aspects, like the conformations of the sugar ring, the base pairing, and so on. This week, we'll look more into the structural aspects and we'll start to relate the structure with the NMR. So, this is already what we have done in the last, discussed in the last class, just a bit more details. So bDNA is the common DNA found in organisms, and normally double helical DNA, it appears like this. So two right-handed helices run in opposite directions. So you can see one helix, if I consider this is 5 prime and up is 3 prime. Then the other strand, which is not shown here, which runs like this, is 5 prime, and here it comes down as 3 prime. So the strands run in opposite directions. Also, the base pairs are very well stacked. So you can see that, suppose, so this is stacking, this is stacking. So the base pairs are kind of well stacked on top of each other. Now in bDNA, two kinds of grooves are formed. One is the minor groove, you can see here with a small indent, and another is the major groove with a large indent. These grooves can have different kind of interactions with different molecules. We also talked about zDNA in the previous class, which is the left-handed helix. So zDNA basically uh, is composed of two left-handed helices, and it is much less symmetric. You can see it is kind of zigzag. It is not actually a good helical structure. And the base pairs are also very unsymmetric. Like here, the base pair stacking is there. Here, the base pairs are not stacked. So that is the difference between bDNA and zDNA. Just excuse me for 1 second. Right. So I was discussing the structural differences between bDNA and zDNA. And as I said, that bDNA is made up of two right-handed helices, and zDNA, there are left-handed helices. Also, in bDNA has a very symmetric structure where you have alternating major grooves and minor grooves. But zDNA has a very unsymmetric structure where sometimes you can have a section bulging out like this, so, and other times you can have a deep groove. So it is a much less symmetric structure, and the base pair stacking is also not good compared to bDNA, where the base pairs are very nicely stacked on top of each other. There are other details. We don't need to look too much into the detail of that. The few important points to remember is that the number of residues per turn is 10 in bDNA, and in case of zDNA, it is 12. Now, it is important to remember that in bDNA, the monomeric unit is a single nucleotide, but in zDNA, the nucleotide dimer, that is the base pair together, so suppose CG, so CG together forms one monomer of the zDNA. So the monomeric unit of zDNA is actually the base pair. This is because the two strands are not in bDNA, the two strands are very symmetrically kept. So one nucleotide in one strand is enough to be a monomeric unit or repeating unit. But in zDNA, because the orientations of the two strands are different, that is why the monomeric unit is a dimer. Secondly, is that the rise per residue is 3.4 angstrom in bDNA and 3.75 angstrom in zDNA. Now, there's a term called pitch, which refers to the rise per turn of the helix. So, if you have a helical structure, the rise, this rise, which is the rise per turn of the helix, is referred to as the pitch. So you can find the pitch by multiplying the rise per residue into the number of residues per turn. So in case of bDNA, it's 3.4 into 10, so 34 angstrom. And in zDNA, it is 3.75 into 12, so 45 angstrom. There are other things which we don't need to go too much into the detail of. In the previous class, we have already looked at the sugar puckering of C2 prime endo or C3 prime endo. One new term that we will look at in this class is the glycoside torsion angle, that is syn or anti. So I will elaborate that in the next slide. Now, what is this glycosidic torsion angle? It is often called chi. So basically, you have the 5-membered sugar ring, and at this position of the sugar ring, it is attached to a nitrogenous base. Now, suppose you have a 6-membered, this kind of a nitrogenous base where you have a fused 5-membered and 6-membered ring. Then when this hydrogen of the 5-membered ring is in the same side as the oxygen, this oxygen of the sugar ring and the hydrogen of the 5-membered ring of the nitrogenous base is close to each other, that is called the anti-conformation. Now, if you rotate around this bond, and now the hydrogen faces away from the oxygen, this is called the syn conformation. So these are two conformations, syn and anti, And it is called the glycosidic torsion angle because this bond is the glycosidic bond. Now, so here what is being said is that the glycoside torsion angle in case of bDNA, for both G and C, it is anti. But in case of zDNA, guanosine is syn, cytidine is anti. So like I said, zDNA, the two strands are not symmetric. And you can see here as well that the two strands, G and C in the two strands, have different orientations of the glycoside torsion angle. So here the two-dimensional structure is shown. You can imagine this anti-orientation is again shown here in a 3D ball and stick model. So you can see here, this is the hydrogen. So this hydrogen is shown here in a red circle and it is close. So this is the oxygen of the sugar. So it is close to each other. That is your anti-conformation. And if it is comparatively far away from each other, that is your syn conformation, where the hydrogen is now pointing away. Now in the syn conformation, the hydrogen actually comes close to these protons on the sugar ring. So here there can be some interactions. We know that this is something we already saw in the previous class, that when you have the sugar ring, 3 atoms will be in the plane and the 2 other atoms, one is above the plane and one is below the plane. The one above the plane, we call it as endo, and the one below the plane as exo. So in this case, it is a C2 prime endo. So this is the case shown here, where the C2 prime atom is above the plane. And this is the case shown here, where the C3 prime atom is above the plane. So it is C3 prime endo. Now the length of the DNA, if we consider 1 billion base pairs, is of the order of centimeters. But the nucleus of the cell is of the order of microns. So for that, DNA is folded in many ways, and now we will look into that. So basically the nitrogenous base pairs, which we already went into in the previous class, you have the purines, which are adenine and guanine, which are formed of fused 5 and 6 membered rings. And you have the pyrimidines, which are 6 membered rings consisting of thymine, cytosine, and uracil. And we have already discussed that thymine and uracil are interchangeable. In RNA, it occurs as uracil, and in DNA, it occurs as thymine. Now, the common base pairs are the purine-pyrimidine base pairs, more specifically the Watson-Crick base pairs. This is the one we showed in the previous class, where you have A pairing with T or U through 2 hydrogen bonds, and G pairing with C with 3 hydrogen bonds. This is the most common base pair that is formed. But other than this type of base pairing, there are many other kinds of base pairs that can also form. And that is what we will discuss now. So one is the reverse Watson-Crick. In reverse Watson-Crick, what happens? So imagine there is a plane passing through this dotted line and you flip the U 180 degree around this line. So earlier the bond was with this oxygen. Now the bond is with the other oxygen because we have flipped U through 180 degree through this line. And R was down, now R is up. The A orientation remains the same. So this is called reverse Watson-Crick. And even for GC, if you imagine the line here and flip the second residue, that is C by 180 degree. So earlier there were 3 hydrogen bonds from amine, this N, and this carbonyl. And now there are only 2 hydrogen bonds possible through the carbonyl and the N. And R before was pointing down, R is now pointing up. So in reverse Watson-Crick, basically there is just a 180 degree rotation of the second base pair about the axis. There is another kind of base pairing called the Hoogsteen base pair. So in the Hoogsteen base pair, one NH of the 5-membered ring is involved in the hydrogen bond. So here you can see the 5-membered ring is completely pointing away from the base pairs, both in Watson-Crick and in reverse Watson-Crick. So in the purine, only the 6-membered ring is participating in hydrogen bonding. In Hoogsteen, it is both the 6-membered and the 5-membered ring that participates. So you have the purine oriented in such a way that the NH2 of the 6-membered ring and the N of the 5-membered ring participates in hydrogen bonding. Similarly, you can have reverse Hoogsteen. If you carry out a 180-degree rotation around this line, then the R group, which was pointed down, now it points up. And it is kind of just like flipping. So earlier this oxygen was participating, now the lower oxygen is participating in the hydrogen bonding. There is a third kind. So we saw Watson-Crick and we saw Hoogsteen. There is a third kind where it is called a wobble pairing. So normally we expect G to pair with C. And so the A pairing with T and G pairing with C, these are the expected base pairs, and this is observed both in Watson-Crick and in Hoogsteen. In wobble, what happens? The pairing is unexpected. So instead of GC, in wobble, you will observe a GU. So you can see here also the carbonyl, similar to Watson-Crick, where only the 6-membered ring was participating in the hydrogen bonding. In wobble base pairing also, only the 6-membered ring is participating in hydrogen bonding. The only difference is that instead of the expected partnering is between G and C, but in wobble, G and U is observed. This is a much more rare case that happens, but it does happen in some organisms. Then you can have, again, this is another example of wobble because A is supposed to pair with T. But if you have A pairing with C, that is called a wobble base pair. And similarly, for any base pair, you can have its corresponding reverse base pair where you flip the second nitrogenous base by 180 degree and the R group. So R group is basically where the sugar ring is attached. So the sugar ring, instead of being attached down, is attached up. So for any, so this is a GU wobble, you can have a corresponding GU reverse wobble where the U is flipped by 180 degree. And similarly, you can have reverse Hoogsteen and so on. So mainly there are three kinds of base pairings: Watson-Crick, Hoogsteen, and wobble. And for each kind of base pair, you can have the corresponding reverse base pairing where you are simply flipping the second nitrogenous base by 180 degrees. Now, so far we have looked at purine-pyrimidine base pairs. You can also have purine-purine base pairs. So basically you can see that both purine and pyrimidine, they have so many hydrogen donor as well as hydrogen acceptors. That is why purine-purine is also possible. So the system of naming these are very like clear. So here A base pair is forming. We call it a N1 amino. So if you remember the numbering of the purines, N1 is this N in the 6-membered ring. So you can see it is the same amino N that is participating and N1 amino. So N1, I'll just label this as N1. N1 is binding to amino, I will call this A. And again, N1 is binding to amino, I will call this A. You're calling it symmetric because both are similar kinds of hydrogen bonds. Then you can have N7 amino. So this is N7 binding to amino. Again, N7 binding to amino. this is also symmetric. You can have unsymmetric or asymmetric base pairing also, where N, one second, N1 is binding to amino and N7 is binding to amino. So these are two different kinds of hydrogen bond. That is why this is a unsymmetric hydrogen bonding. Then you can have GG base pairing similarly. When G comes into question, now you can have hydrogen bond acceptors as carbonyls as well. So here you have N1 carbonyl. This is N1. This is carbonyl. Again, N1 carbonyl. And this is a symmetric base pairing. Similarly, you can have N3 amino. Then you can have unsymmetric base pairing between G and G with N7 to N1. So here in this case, N1 is here, N7 is here, and you have carbonyl to amino. So you can see the name here. It is N1 bonding to N7. That is what is here. And carbonyl bonding to amino. Carbonyl and amino. So basically the names are all based on what kind of hydrogen bonds are forming. So these are all homopurine base pairs where both the purines are same. So it's AA or GG. You can also have heteropurine base pairs where the two purines that are binding are different. So since you only have two purines anyway, so the heteropurine base pair can only be GA or AG. But here also the naming is similar. So you can have N1 carbonyl amino. So this is N1 of guanine, this is N1 of adenine, this is carbonyl, this is amino. So again, the names are based on the kind of hydrogen bond that is forming. And these are the different kinds of heteropurine base pairs. So similarly to heteropurine, you can have pyrimidine-pyrimidine base pairs. And these can be homopyrimidine. So you can have two of the same pyrimidines like CC or UU. Or you can have heteropyrimidine where the two pyrimidines are different. So CU, UC, and so on. We don't need to go too much into the details of this. What you can do is just remember the numbering, because if you remember the numbering, you will automatically be able to name. So if a structure is given and you are asked that what should be the name of this structure, if you just remember the numbering of both the pyrimidine and the purine, you will be able to derive this name. So you don't have to remember each of the base pairs separately. Just remember the numbering. Now there is another kind of base pairing possible, which is called the I motif. So this forms, here it is shown between C and C+. C plus is basically a cytosine which has been protonated, so it gets a positive charge. So you can see cytosine here. So this N is attached to your sugar ring. So cytosine only has one NH2 group. Now, if this N also gets protonated by an H, it forms a C+. That is what has happened here. You have your NH2 group here, and this is the extra proton that is added. And from here, it is attached to the sugar. So because of this extra proton, it is now C+. And if you look at the normal CC, you can see the CC homopurine base pairing. There are two hydrogen bonds between them. Or another kind of CC also has two hydrogen bonds between them. But if you add an extra hydrogen here, then the number of hydrogen bonds, so this can also form a hydrogen bond now, and the number of hydrogen bonds becomes 3. So now the pairing is even stronger. That is what is shown here. Due to the addition of this extra hydrogen, 3 hydrogen bonds are forming, and so C plus is stronger than CC because of the presence of this third hydrogen bond. So now what can happen is that we will discuss these kind of structures in the upcoming slides, where basically multiple base pairs can stack on top of each other. So that I'll come to in the next slide. So far we have seen this kind of structure, which is the normal duplex, where there are two strands of nucleotides and there is base pairing between them. Now, when the DNA needs to express itself to form proteins or for replication, the double-stranded structure needs to open up. In that case, sometimes regions like loops can form, where in the loop region it is single strand. Now, this loop region can now form mRNA, or it can be used for replication of only this part of the DNA. So, for any activity of the DNA, it needs to become a single strand. And that can happen in this kind of loop region. If you have a structure like this, it is often called a hairpin, where you have a duplex strand where there is double-stranded DNA called the hairpin stem, and you have a single-stranded loop called the hairpin loop. Other kinds of structures are bulges. So bulges are basically where you have loops, but so here the loop is formed at the end of the duplex strand. If it is formed on one side of the duplex strand, it is called a bulge. And so you can have a bulge formed of multiple nucleotides. You can see that each unpaired base is shown like this. Or you can have a single base bulge where there is only one point, that is one base, that is unpaired. So depending on the sequence or what is the function of the sequence, you can have different kinds of bulges forming. Then you can have what is called a mismatch. So basically, if the two strands only if they are complementary, that is A with T and C with G, you will have a proper double-stranded DNA. But somewhere between the strands, if it becomes non-complementary, then the base pairs will not pair with each other. So there are two unpaired base pairs, because suppose this is A and G. They can't pair with each other, so they will just point away and form what is called a mismatch. You can have this mismatching section be symmetric. So if there are three unpaired bases on this side, there are also three unpaired bases on this side. That is symmetric. If there are three unpaired bases on this side, but there's only one on this side, that is asymmetric. And these are kind of called internal loops because they are formed within the double-stranded DNA. You can also have structures where there are, so two double-stranded DNA are joining, and at the point of joining, there is a bulge or single-stranded region. Then it is called three-stem. It's considered as 1, 2, and 3. You can also have four stems, that is four double-stranded DNAs coming together, and wherever they are joining, there can be single-stranded bulging regions. So these kind of structures are generally found in ribosomal RNA or tRNA, and we are not going too much into the details of that. So far we have seen base pairing between two nitrogenous bases, which are often called duplexes. We can also have triples where three bases come together. So you can see here between A and T, so this is the Watson-Crick base pairing that is formed between this A and this T. You can also have a T coming from the other side and forming the Hoogsteen hydrogen bonding. So between this T and this A is the Hoogsteen hydrogen bonding. So because the Hoogsteen hydrogen bonding forms through the 5-membered ring of the purine, and the Watson-Crick hydrogen bonding forms from the the Hoogsteen hydrogen bonding forms both from the 5-membered and the 6-membered ring, and the Watson-Crick only forms from the 6-membered ring. Both can happen at the same time in order to form what is called a triplex. Now, this can also, like this is a TAT triplex. You can also have CGC GGC, or AAT. Different kinds of triplexes are possible. And what these arrows indicate is that you remember studying about nosy, where when two protons are less than 5 angstrom distance from each other, then the two protons will show a peak in the nosy spectra. Here, the protons which are close to each other in space are indicated by these arrows. That is, we expect to see interproton, like between these two protons, we expect to see a peak in the nosy spectra. Similarly, for the other arrows shown here. So these are the peaks observable in nosy, and from the nosy peaks, we already know we can calculate the distance. So nosy directly proportional to 1 by r to the power 6. So from the intensity of the nosy peak, we can calculate the interatomic distance and find the structure accordingly. Now, when you have, we know that when there is a base pairing between two nitrogenous bases, the double helical structure forms. But when there is this kind of three nitrogenous bases or triplex forming, the triple helical structure is formed. Basically, you can imagine a double helix formed by the two green strands, and you have a third strand, which is red in color here, that inserts between the two strands of the double helix. So this is how the triple helical strand looks like. You can also have quadruplex, where four nucleotides come close to each other and bond. So this is a very stable structure called the G tetrad or G quadruplex, where you have four G nitrogenous bases come close to each other and hydrogen bond in this kind of a fashion. So you can see it is a very symmetric structure. Each G has two hydrogen bond acceptors, that is this N of the 5-membered ring and the carbonyl of the 6-membered ring. And each G also has two hydrogen bond donors, that is this amino and this iminoproton. This is true for all four Gs, and together a very symmetric and stable structure is formed. Now, whenever you have this kind of quadruplex formation, different quadruplexes can stack on top of each other in different strands. So there can be different arrangements of the quadruplexes, and that also we will look into a little bit later. So now we have briefly looked into the structures that are possible. We will now start to relate the structure with the NMR. So in 1H NMR, we know already that it ranges from 0 to around 15 ppm. Now, DNA here is shown in blue and RNA is shown in red. Now, in DNA, you have a deoxyribose sugar, and in RNA, you have a ribose sugar. So protons 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime are all part of this sugar, and they appeared in this range, which is from here around 2 to around 6.57. So the difference between DNA and RNA is that in DNA, it is mainly this 2 prime position. So in DNA, the 2 prime position has two protons. We call one as 2 prime and 2 double prime. You can see here it's H2 prime and H2 double prime in case of DNA. In RNA, the 2 prime position has one proton and one OH group. So we know OH has an electron withdrawing effect, and because of the electron withdrawing effect of the OH, for this hydrogen, the delta will increase. Indeed, we see in RNA, the H2 prime is very much downfield shifted. Compared to DNA, the H2 prime in RNA is downfield shifted. And you also have only one H2 prime because in the other position, there is a OH. That is the main difference in the sugar ring regions. So because of this difference in H2 prime in DNA and RNA, the sugar ring protons in RNA get very much crowded in the same position, but in DNA, they are quite separated. Because if you see in DNA, the 1 prime, 3 prime, 4 prime, 5 prime. In all of this, there is an electron withdrawing OH group. So other than 2 prime, all the other protons are quite downfield shifted. But in 2 prime and 2 double prime, there is no OH group directly attached, and so they are upfield. So in DNA, the sugar ring protons are quite spread out. As we move more downfield, we will next observe the nitrogenous base protons. So the H2, H8, and H6 appear next around 7 to 9 or 8.5. So basically your H2, 6, and 8. So 6, yeah, these are, you can see the proton labelings here according to the numbers that are shown here. So these are the protons of the nitrogenous bases and they appear next. Now, this is the region where the amino protons appear. So you can see H6 is basically this amino proton, which appears in adenine. H2 is this amino proton, which appears in guanine. And similarly for H8. So yeah, so H8 can is referred to here. And so you have the different amino protons of G, C, and A. So this numbering is a bit wrong. This is actually H8. So the number here is 8. You can understand it when it pairs with the sugar ring. This is what they are calling as H8. So here is where the amino protons are appearing. Now, often the amino protons are not observable in the spectra because they exchange with water. They exchange. So suppose you're taking the spectra in D2O. You have the amino protons and you have D2O. Now, amino protons can exchange with D2O to give you and we know deuterium is not NMR active. So then you will not be, so this is NMR inactive. You will not see any NMR for this. So if you take the spectra in D2O, often you will not be able to see the amino protons because they are exchangeable protons. So often in order to see the amino protons, the spectra has to be taken in H2O. As you go more downfield, you will see the iminoprotons, that is the NH protons. So you can see the different NH protons, which are the iminoprotons. And they appear more downfield. Now, these are not that much exchangeable. So these you will normally be able to see. And also iminoprotons are often involved in hydrogen bonding. Amino proton, although they are involved in hydrogen bonding, one amino proton is always free. So if you look at the hydrogen bonding here, you can see this amino proton is free. This is involved in hydrogen bonding. So the free amino proton can always exchange with the solvent and hence it will disappear if the solvent is D2. Also, the different base pairs like AT, GC, or in case of RNA, AU and GC, they will also appear in this range. And if you have loops and other structures, they also appear in this range from 10 to 15. So we will look more into that later, but these are the basic ranges of the protons and nucleic acids that appeared in 1H NMR. Now we will look at an NMR, 1H NMR of nucleic acids. Here you have a sugar ring and because it's DNA, you have 2 prime, 2 double prime and 1 prime and 3 prime, 4 prime, again 5 prime, 5 double prime. And in case of the bases, you have H2, H5, H6 and H8. You have different hydrogens of the bases that will be observable. So one very distinct thing is the CH3 hydrogens from thymine. Thymine is the only group, CH3 group, in all the nitrogenous bases, and it appears at a very distinct chemical shift that is less than 2 ppm. In this sequence that is shown here, you have 2 and 3, 4 thymine. So if you integrate the CH3 peaks here, they will, if you integrate these peaks, they will come to 4 hydrogens. Sorry, 4 CH3 groups. So 4 into 3, 12 hydrogens. So this is a very distinct chemical shift, and it helps to understand how many thymines there are. Other than that, you can see the 2 prime are compared to the other protons in the sugar ring, the 2 prime are most upfield because there is no OH directly attached. And then you have the other protons like 4 prime, 5 prime, which are overlapping very much. You can see that it is almost impossible to find out the sequence from just 1H NMR because there is so much overlap. And of course, as you go more downfield, you get the protons of the nitrogenous bases. But the point of this is that just from 1H, it is not possible to figure out the spectra. So here we were talking about the non-exchangeable protons, which are the protons of the sugar ring and the nitrogenous base. Now we will look at the exchangeable protons. So here we have a duplex sequence. So GGAA, GATC, TTCC. This is the duplex segment. And at both ends of the duplex, you have a loop made up of 4 cytosines. So these cytosines that are part of the loop, they are not paired. And these will not be observable because they will exchange protons. So first of all, we are taking this spectra in water. So the cytosines will be able to exchange protons with the water. And we know we have seen that NMRs for protons which are very much exchangeable, like carboxylic acids or alcohols, they are not often observed because if the exchange rate is too high. So I was talking about this exchange. Now, even instead of D2O, if it is H2O and the exchange is too fast, then also the proton will not be able to be detected. So that is what they are saying here, because in the single stranded loop region, it is exposed to the water. The exchange will be so fast that the protons of these cytosine you will not observe. Other than that, if you look at the base pairs that are forming. So here the normal Watson-Crick base pairs are forming AT and GC. Now for each base pair, there is only one iminoproton. So you can see that in this base pair also there is one iminoproton, and in this base pair also there is only one iminoproton. So we are now looking at the region of the iminoprotons. Why not the amino? Because we have already discussed that aminoprotons are not that much reliable because they are very much exchangeable. So instead of looking at the aminoprotons, we are directly looking at the region of the iminoprotons that are 12 to 15 ppm. So when we look at this 12 to 15 ppm region, we will be able to see these iminoprotons. So number of so for each base pair, we will see only one iminoproton. Now about this sequence, one special thing is that it is a self-complementary sequence. So if you read the sequence from this side and you read the lower strand from this side, they are the same. So there is a kind of symmetric nature of this sequence. So how many iminoprotons are we expected to see? We will see this GC, this AT, this AT, and this GC. And one more AT we will see. Because from the middle of the molecule, again, it becomes symmetric. So if you, so GC, GC, AT, AT, GC, AT. And if you read the other end of the molecule from down, it is the same, GC, GC, AT, AT, GC, AT. So there are only 6 unique base. So there are only 6 base pairs that are unique. These are unique. So you will see 6 iminoprotons. There should be 6 peaks in the iminoproton region. Indeed, you can see the peaks have been labeled as G. So these are the G peaks. G peaks meaning for the CG base pair. So the CG base pair will have 1 iminoproton due to the guanine. These are the 3 peaks of the CG base pairs. And there are 3 peaks of the AT base pairs. They are written as the T peaks because the iminoproton is on thymine. So because there are only 6 base pairs that are unique, the other 6 base pairs are the same if you just read from the opposite direction. So basically it's a symmetric molecule. So you will only see 6 base pairs. That is, you will see 6 iminoprotons. And indeed 6 iminoprotons are seen. Now, as you increase the temperature, the peaks start to merge. Like you can see in this temperature, the 3 thymine AT base pair peaks are clearly distinguishable. But as you come to this temperature, which is higher, all the 3 peaks merge. So with increase in temperature, what happens is that the base pair starts to separate. As you increase the temperature, there is a melting of DNA, and adenine and thymine, the base pair starts to break up. As the base pair starts to break up, the iminoproton gets exposed to the solvent and starts exchanging with the solvent, and then the iminoproton peak will disappear. Like I said that for the cytosine in the loops in which already the iminoprotons are exposed to the solvent, you don't see any peak. So similarly, if you continue heating, because of the breaking of the base pairs, the iminoprotons will get exposed to the environment, the solvent environment, and the peak will disappear. Now, we will look at a DNA hairpin. So hairpin we already saw is basically you have a section of a duplex DNA and you have a loop section. Now the nucleotides in the loop, again, they will not be observable as they are exchanging with water. We have already seen that nucleotides in the cytidine loop are not observable. So similarly here, however many nucleotides are there in the loop, they are all unpaired and so they are exposed to the water and so they are not observable. Other than that, for each base pair, you will see 1 imino peak. So here there is no symmetry. Here again, the symmetry was because the loop is 4 cytosine on both sides. So whether you're reading from this side or this side, because the loops are symmetrical, there was a symmetry. But here it is clearly not symmetric because you have loop on only one side. So if you just look at the sequence, it appears to be symmetric because you have GAA-TTC. And again, if you read the lower strand from the opposite side, you have GAA-TTC. But still it is not symmetric because you have loop on only one side. So in this case, all the 6 base pairs are clearly distinguishable, and you will see for 6 base pairs, you will see 6 iminoprotons. Indeed, you can see in the imino region, the peaks are labeled as 1, 2, 3, 4, 5, and 6 because you are getting 6 peaks from the 6 iminoprotons. And as the temperature is increasing, the peaks start to merge as discussed before. So as I said, the melting of the DNA duplex. So when you have, so this is what is shown here again. Sorry, this is of another sequence. Now, as the temperature is increasing, the base pairs start to separate because of breaking of the hydrogen bonds. And so the iminoprotons in the base pair will get exposed to the solvent and the signal will start disappearing. So that is what is happening here. If you just look here at the lowest temperature shown, there are 4 Cg base pairs and 2 AT base pairs. Now, as you increase the signal, the Cg1 is the first to disappear. You can see Cg, I'll just label this 2, 3 and 4. Cg2, 3 and 4 are still there, but Cg1 has disappeared from 0 to 20 degrees Celsius. So we know that the exposed, so Cg1 is the one that must be at the end of the sequence or the terminal. Because any nucleotide, so as we go from the terminal to the internal region of the nucleotide, the stability of the base pair increases. Because the terminal nucleotides are already exposed to the solvent, the stability is not that great. And so as you heat the DNA, the terminal nucleotide is the first, the terminal base pair is the first to disappear. So you can see initially Cg1 disappears. So that must be the terminal base pairs. Eventually, as you go on, Cg2 will be the next to disappear. So that must be the next in the sequence. So if you look at the order of disappearance, 1 will disappear first, then 2, then 3, then 4. So like this, we will be able to predict that which is the base pair that is near the terminal and which are the base pairs that are towards the interior of the duplex. So another thing we can study is that suppose we have a modification in the DNA. This is the general DNA structure where 2 deoxyribose sugars are attached by a phosphodiester bond. Now, if one of the oxygens in the phosphodiester bond is replaced by sulfur, that is called a phosphothioate bond. That is the first. So you have 3 DNA here. First is the unmodified DNA. Another is the one where one oxygen has been replaced by sulfur. And the second modification is where this oxygen has also been replaced by sulfur. So you can see it is written as 2s because both oxygens have been replaced by sulfur. And this is 1s because one oxygen has been replaced by sulfur. So then if you plot the absorbance versus the temperature, they will give this kind of sigmoidal curve. And in order to find the melting temperature, that is the temperature at which the DNA duplex starts breaking, you have to take the midpoint of this sigmoidal curve and draw a normal. So you can see for the unmodified curve, which is the lowest one, the melting temperature is around 60 degrees Celsius. For the monosulfur derivative, the melting temperature is around 45. And for the disulfur derivative, the melting temperature is around 20. So as you go from unmodified to monosulfur to disulfur, the DNA is becoming more and more unstable because the melting temperature is decreasing. So using this, we can also predict that whether the DNA is stabilized or destabilized by a particular modification. So now we have looked at several kinds of 1D NMR, but we have also seen how very much crowded the 1H NMR is. So 1H NMR on its own is not very useful because of the crowding. So we will now look at the 2D NMR or NOSI. So in the 2D NMR, again, NOSI basically in both the axes, you will have 1H NMR. So as discussed before, the CH3 protons of thymine will be the most upfield shifted. Then you will have the 2' 2" of the DNA, followed by the other sugar protons. And then you will have the different protons of the nitrogenous base. So if you look at each section of the NOSI spectra, so this section is an interaction between the H2H6H8 of the nitrogenous base, and similar because this is on the diagonal line. So this section is basically depicting the interactions between the base protons, or it is depicting base-base stacking. This section, if you see, is between the protons of the nitrogenous base and the 3' 4. So we can just draw, like, we can see it is between the protons of the nitrogenous base H2, 6 and 8, and between 5' 3' 4' of the sugar residue. Similarly, this section is between the protons of the nitrogenous base and the methyl and 2 of the sugar residue. So we can take each section of the nosy in order to find out that what are the correlations between protons that are close to each other in space. And from the nosy intensity, we can find the interproton distance. Here is basically, it is shown two residues are shown. So you have the ith residue. Here is the sugar ring attached to the nitrogenous base, and you have a phosphodiester bond attaching to the I plus first residue. Again, there's a sugar ring, there is a nitrogenous base. Now, basically the noses that are expected are shown. You have intra-residue interactions, that is between the base and the sugar of the same residue. So in the ith residue, you have an interaction between H1 prime and this H8, which is, if you take the nosy intensity and find the distance, it comes around 3.5 angstrom. Similarly, for the I plus first residue, you have an intra-residue interaction that is within the same residue between H1 prime of the sugar ring and again H8. Again, the distance is similar. The more interesting nosy is between the two residues, or inter-residue interaction. So basically, the H1 prime shows interaction with this hydrogen of the nitrogenous base, and this is called inter-residue because it is between two residues. So the sugar proton will interact with the nitrogenous base at the I plus first end. The nitrogenous base of the I minus first end is much farther away. So the interaction will only be from the H1 prime of the sugar at the 5' end to the H of the nitrogenous base at the 3' end. So it is a sequential interaction. So I will interact with I plus 1, I plus 1 with I plus 2, and so on. So from the nosy intensity, these distances can be derived. So now we have a closer look at the nosy. So we already know that from the nosy, we can find out sequential connectivities and we can predict the sequence. So each nitrogenous base will have two cross peaks. We have seen here also. So each nitrogenous base has two cross peak, one with the sugar of the same residue. So imagine this I plus first residue. This nitrogenous base has two cross peaks, one with the H1 prime sugar of the same residue, one with the H1 prime sugar of the previous residue. So there is one self peak which is shown in green and one sequential peak or cross peak shown in red. So imagine we start from here, which is T8. We are starting from the 3' end. So if we start from here, we can see that for time T8, this is the self peak of T8, that is T8 interacting with itself. As we move down, we will interact a sequential. So what is shown in green and red here, basically it means they are antiphase. So as we move in the same line, we will encounter a peak of the opposite phase, which is a cross peak. So this means that this is of the previous residue. So basically we are moving in this direction. We are moving from 3 prime to the 5 prime end. So we are starting from the T8, this thymine, and as we move down, we will encounter a cross peak. In order to find which residue this cross peak belongs to, we move horizontally and we encounter G7. So we will know that the seventh residue is G. Then we will move to the next. So this is from the self peak, we know it is G. Then from here, we will move till we find another cross peak. And as soon as we encounter the cross peak, we can move horizontally and find the self peak. And the self peak will tell us this is another G. So similarly, you can move through the entire sequence. So from G6, you will get G5, from G5, C4, then G3, then G2, and finally T1. Now T1 is the 5 prime end. So T1 will only show self peak because there is no previous residue with which it can interact. So if you look at T1, there is only one peak in T1, this green peak or the self peak, and that will tell us we have reached the end of the nucleotide. So basically, nosy correlations like this help to find the sequence of the nucleotide. Just give me 2 minutes. Right. So we will now look at the sugar puckering. These are already things we have discussed in the previous class, where we know that there is a pseudo rotation of the sugar ring. Basically, you have a 360-degree rotation, and so if you consider the middle of this 360-degree, all angles in the upper position are referred to as north. And basically in north, the 3 prime atom is above or endo, and the 2 prime atom is below or exo. And any orientations to the south of this line are referred to as south. And in that, you have the 2 prime above or endo and the 3 prime below or exo. So this we had already discussed in the previous class. Now we will correlate this angle with the J coupling constant. Now in a sugar ring, you can have coupling between 1 prime, 2 prime, 1 prime, 2 double prime. So this is 2 prime, 2 double prime. Then you can have 2 prime, 3 prime, 2 double prime, 3 prime. And you can have 3 prime, 4 prime. These are all the possible 3J couplings possible. Now, basically what is known is that as you vary the pseudo-rotation angle, this is how the coupling constants vary. So these 5 coupling constants are shown in the graph here, and each of them vary in different ways. So first we will look at the C2 prime endo structure. In the C2 prime endo, the pseudo-rotation angle is around 144 degree. So at 144 degree, we have drawn this kind of a normal. So wherever this normal is cutting these graphs is the value of J that you will have. So if you find the value of J like this, you will find that the J 1 prime 2 prime. So between 1 prime and 2 prime, which is this curve, it comes around 10 hertz. Then between 1 prime and 2 double prime. So imagine that this is 2 prime, this is 2 double prime. So 1 prime with 2 prime is around 10 hertz. 1 prime with 2 double prime is around 6 hertz. So similarly, if you find out where are the places, this line, wherever it is cutting these graphs, you will find and you draw lines to the x-axis. So 1 prime 2 prime is 10 Hz. 1 prime 2 double prime is around 6 Hz. Similarly, if you find out the other coupling constants, they are listed here. It is the interesting part is that 2 double prime 3 prime, which is this dotted line, and 3 prime 4 prime, which is this line, are very low. They are like 1.2 to 2, 1 to 2 hertz or 0 hertz. So these coupling constants are very low, and normally this we will it is not observed. Such low coupling constant in 2D spectra, it is not able to be differentiated. Now, if you look at the 3 prime endo structure, which is an angle of you can see the 3 prime endo has the pseudo rotation angle 0 to 18 degrees. So again, we have drawn a normal here. And if you calculate at this C3 prime endo structure, where we have taken the pseudo rotation angle to be somewhere between 0 and 18 degrees, you can see in this case, the 1 prime 2 prime coupling is 0 hertz and all others are greater than 6 hertz. I'll just discard this. So you imagine this line. So pseudo rotation 0 to 18, we have drawn this line, and you can see this, which is 1 prime to 2 prime is 0 hertz, but all others are coming somewhere from 6 to 10 hertz. So that is the difference between the coupling constants of the C2 prime endo and the C3 prime endo structures. Now, what happens to the cozy peaks? In the cozy peaks, if you look let us first look at C3 prime endo. In C3 prime endo, we just saw that 1 prime 2 prime coupling constant is 0 and all other coupling constant is greater than 6 hertz. So if you have the peaks H1 prime, 2 prime, 2 double prime, 3 prime, 4 prime written here, similarly in the x-axis, you will find 4 peaks. You will find uh, these are the peaks that you will observe. 1 prime with 2 double prime, 2 prime with 3 prime, 2 double prime with 3 prime, 3 prime to 4 prime. What peak you will not observe is the 1 prime 2 prime peak, because we already know the coupling constant is 0. So this cross peak will not be observed in the cozy. And in case of C2 prime endo, you have two coupling constants which are very low, the 3 prime 4 prime and the 2 double prime 3 prime. Both these coupling constants are very low, and so both these peaks will not be observed. So only 3 peaks are observed in the C2 prime endo in the cozy spectra. So now we will look at the fine structure of these peaks. So fine structure of cozy is something we have already studied before. So suppose fine structure is basically we expand each of these peaks and we see what is the in phase, out of phase, and so on components of the spectra. So suppose we are looking at C2 prime endo and we are looking at the interaction between 1 prime and 2 double prime. So we know that firstly, we know that 1 prime is split by two things. 1 prime has coupling with 2 prime and with 2 double prime. And we know that the coupling with 1 prime 2 prime is around 6 hertz, and 1 prime 2 double prime is 6 hertz, and 1 prime 2 prime is 10 hertz. And we also know that active coupling is the one we are looking at. So right now we are looking at 1 prime 2 double prime coupling. So initially the 1 prime is split by 2 double prime into 2 components. And because 1 prime 2 double prime is the active coupling, that is the coupling we are looking at, the components will be of opposite phase. So it will be plus minus. The next splitting of 1 prime is due to 2 prime. Now it is important to remember that the 1 prime 2 prime coupling is greater. It is 10 hertz. So compared to this distance, which was 6 hertz, this distance is 10 hertz. So when you have the second coupling as greater, the lines will cross each other like this. And because 1 prime 2 prime is passive coupling, we are not looking at it directly, the splitting will have the same sign. So plus will split into plus, minus will split into minus. So the components of 1 prime are plus minus plus minus. That is what is written here. Now 2 double prime, it will combine with 1 prime and it will also combine with 2 single prime. Now, if you remember, we just saw that 2 double prime, 3 prime coupling is very small. It is around 0 Hertz, so we ignored it. So 2 double prime is initially split by 1 prime, and this is 6 Hertz. Then we have 2 double prime split by 2 single prime, and that is geminal coupling, and it is huge. It is around 14 Hertz. Now, so because it is huge, again, the two lines will cross each other. The first one was active coupling. So the two signs are different, plus minus. And then we have two passive couplings. So plus splits into plus and minus splits into minus. So the fine structure of H2 double prime is plus minus plus minus. That is what you have written here. So now you can multiply this to find the different signs of the fine structure. And basically you have 4 into 4, 16 components of this peak. We can also look, so now we have looked at 1 prime, 2 double prime. We can also look at 1 prime, 2 prime. So 1 prime earlier, so 1 prime is being split by 2 prime and 2 double prime. Because we are looking at 1 prime, 2 prime, this is your active splitting and this is your passive splitting. So first we will do the active splitting. The 1 prime 2 prime splitting is happening first and it is 10 Hertz. And because it is active, it is splitting into plus minus. Then you have 1 prime 2 double prime splitting, which is 6 Hertz. So the second splitting now is smaller. So there is no crossing of the lines. In the previous case, the second splitting was larger. So the two components were crossing each other. But now, because the second splitting is smaller, it is only 6 Hertz, the components do not cross each other. And because it is passive splitting, plus remains as plus and minus also splits into minus. So now the 1 prime component is plus plus minus minus. That is what is written here, plus plus minus minus. Now 2 prime is being split by 1 prime, 2 double prime, and by 3 prime. So it is being split by 3 things. So first we will look at the largest, that is 2 prime 2 double prime. So in these 2 prime 1 prime is active. This is passive. This is also passive. We will look at the largest coupling constant, that is the geminal coupling constant 2 prime 2 double prime first. So first 2 prime is splitting into two components by 2 double prime. And this is the largest splitting constant, that is geminal coupling at 14 Hertz. But this is passive coupling, so the signs remain as plus plus. Then we will see the 2 prime 1 prime splitting. 2 prime 1 prime splitting is less, so it is 10 Hertz. So initial splitting was 14 Hertz, now it is 10 Hertz. So now there is no crossing because the second splitting is lower. But 2 prime 1 prime is the active coupling. So plus, because it is active, it will split into two components of opposite sign. So plus will become plus minus, plus will become plus minus. Finally, the lowest coupling constant in the 3 here, 2 prime 3 prime is happening. So each component will again split into two components because of the 2 prime 3 prime splitting. And because these two components are so close to each other, when they split, they will overlap each other. This 2 prime 3 prime splitting is also passive coupling. So plus gives rise to plus, minus gives rise to minus minus, plus gives rise to plus plus, and minus gives rise to minus minus. So this is the order of the signs in the C2 prime. And now you have 8 components. So you have 8 into 4, that is 32 components of this peak. So this is the fine structure of the 1 prime 2 prime peak. Now when you have so many components, many of the peaks will cancel out. Here you have the cozy spectra shown, where many of the peaks are cancelling out. You can see that. So this is 1 prime 2 prime. 1 prime 2 prime is expected to have 16 components. But so we are looking at C2 prime endo, which is around 144 degree. So it is expected to have 16 components, but you can only see 4 components. So many of the peaks are cancelling out. Similarly, this is 1 prime 2 double prime, and we are looking at 2 prime endo, that is 144 degree of the pseudo rotation angle. So this is expected to have 32 components, but here you can only see 16 components, and that also the components in the middle are cancelling out. So because of this, some modifications are being done to the cozy pulse program so that the resolution is better. One of these modifications is called cozy 45 or e-cozy. Basically, in cozy, you have a 90-degree pulse, then you have a T1, then you have another 90-degree pulse, and you collect the FID at T2. But instead of the second 90-degree pulse, if you put a 45-degree pulse or a smaller pulse, like even 30 degrees, then it is called a cozy 45 or e-cozy. What this does is that the intensity in the cross-peak fine structure is altered so that the fine structure is improved. The cancellations that were happening between the fine structure of opposite signs, the cancellation is reduced. And if the cancellation is reduced, we are able to find more coupling constants. So we know that from the fine structure, we can find coupling constants, but it is only when we can see the fine structure in good resolution. So this is one of the techniques used to improve the resolution of the fine structure. You can see a comparison here. You have cozy on the left hand side and cozy 45 on the right hand side. First is between 1 prime and 2 prime. We again look at the C2 prime endo, that is the 144 degrees. You can see that in cozy only 4 peaks were observed. But in cozy 45, 7 peaks are observed. So the number of peaks are very much increased. The fine structure is much more resolved, and this will help us to find more of the coupling constants. Similarly, you can see between 1 prime and 2 double prime. We again look at the C2 prime endo region. You can see, although there are many peaks here, they are not well resolved. The peaks in the middle are all getting cancelled out. But in case of cozy 45, there are 8. So there are 8 peaks. The middle peak is kind of merging. There are 8 very clearly defined peaks. So it is very easy to calculate the coupling constant in this. So it is not always that the number of cross peaks will be increased. You can see seemingly here in cozy, the number of cross peaks is higher, but the resolution is so poor, it is not possible to calculate the J coupling constants in this way. But in cozy 45, although the number of the fine structure is reduced, it is so well resolved that the J values can very easily be calculated. So the main goal of this cozy 45 or e-cozy is to simplify the fine structure and give it better resolution so that the J coupling constants can be calculated. You can see the example here. One example is shown where you have H1 prime here, and in the other axis you have H2 prime and 2 double prime. So in this case, you can find this is an example from cozy 45. So the resolution is very good. If you take the difference, so between this peak and this peak, this is the coupling constant between 1 prime and 2 double prime. This 1 prime and 2 double prime coupling constant, which is coming around 5.8 Hertz. And if you take another similar distance in the lower peak, you will get 1 prime, 2 prime coupling constant, which is around 9.5 hertz. And both of these will have the geminal coupling constant, that is, so 2 prime with 2 double prime, this is giving you 14.1 hertz, and that will be the same here as well, 14.1 hertz. So because the resolution is so good, it is very easy just to find the coupling constants between the different protons from the COSY 45 experiment. Now, another use of this kind of experiment is that you can see the COSY NMR of a DNA duplex before and after it binds with a certain drug. Here it is mitramycin. So before binding with the drug, what is the COSY? And after binding with the drug, what is the COSY? That will help you understand whether there is a change in the sugar geometry or other conformation upon binding of the drug. So that is just one of the applications. We are not going into the details right now. So we saw that one of the ways to improve the resolution or simplify the fine structure is called as COSY 45 or E-COSY. Another technique is the constant time COSY or CT COSY. We have studied this before. Basically, in CT COSY, what happens? There is decoupling along the F1 axis. So you can see that in COSY, you have splitting both along x-axis and along y-axis. In CT COSY, what happens? This splitting is removed. So you have splitting only along x-axis. So this helps to simplify the structure. So you can see here 1 prime, 2 prime, COSY is shown. On the left hand side, it is COSY. On the right hand side, it is CT COSY. So suppose we look at this structure where we are supposed to get 2 coupling constants, but the middle peaks cancel out so much, you are only able to get 1 coupling constant. In this case, because you are decoupling along the x-axis, you can actually get 2 coupling constants, one here and one here. So CT COSY is again another method to simplify the fine structure. No information is lost because you have your delta informations on this axis. This axis basically has your delta as well as J information. And this axis has only delta information. So nothing is being lost. And in this case, you have delta plus J in both the axes. So nothing is being lost. You are just simplifying the fine structure. Another example is between 1 prime, 2 double prime. You can see that COSY, it is because there are so many peaks in the fine structure, the many peaks are getting cancelled out. But when you do CT COSY, you can very easily derive the J coupling constants. Because of the decoupling along one axis, the cancellations are reduced and the resolution is the J values from the CT COSY. Now, there is another angle that is important. That is the epsilon angle shown. So you can see this torsion angle between the C3 prime and O3 prime, that is the epsilon angle, is important. It is the angle connecting the connecting the sugar to the next residue through the phosphodiester bond. Epsilon is the rotation along the C3 prime O3 prime bond. This epsilon value is very restricted or constrained. It is around 270 degrees. So this is a simulated curve where the epsilon angle has been varied from 0 to 360 degrees. And it is seen that around 270 degrees, the structure, 270 degree value of epsilon, the DNA structure has the lowest energy. But as you increase the change the value of epsilon, the energy increases very rapidly. So epsilon can only exist in a very narrow range. So probably in just a small range here, and in a small range here, epsilon can exist. Because anywhere outside of this range, the energy becomes so high, it becomes so unfavorable that the DNA structure cannot form. So this is another one of the constraints given for structure optimization. So now we will look at structure optimization of the DNA. So what are the constraints that we have? First, we have distance constraints. So from nosy cross-peak intensity, we can obtain a distance range. So our expected distance will lie between an upper range RU and a lower range RL. This range will be obtained from the nosy cross-peak intensity, and this is what we call the distance restraint. Then we have an energy function. Energy function has two components. One which is EF, that is the intrinsic energy. That means the energy due to the bond distance, bond angles, torsional angles, and so on. And the second portion is due to, it's called ENOE, or the energy due to NOE potential. Basically, the formula for this ENOE is that you take the deviation of the distance from the lower limit and you square it and multiply it with the force constant, and the deviation of the distance between two protons with the upper limit, again square it, multiply with the force constant. This is done for every pair of protons between which we see the NOE. This is your ENOE. Now, if this distance condition is satisfied, the ENOE will turn out to be 0. So basically, in any algorithm or optimization program, what happens is that the program keeps optimizing the structure till the ENOE becomes 0. Because when the ENOE becomes 0, it means that all the distance restraint is satisfied. That is, all the data we got from the NOE is being satisfied. So that is the basic concept by which all these optimization algorithms are developed. One of these algorithms is called torsion angle based distance geometry, or TAND algorithm. So you have an example is shown here where you are starting. So before I go into that, just 1 second. Right. So before I go into that, a very like common concept is that for any structure, to uniquely define the structure, there are a certain number of distances that you have to define. So suppose you want to uniquely define a triangle, you will need to define the distances of all three sides. Or to uniquely define a quadrilateral, you have to define the distances of all four sides. So similarly, there are a certain number of minimum distance constraints that are needed to uniquely define the DNA structure. Suppose we start from a 12-mer DNA and we start from any random geometry. We start the optimization process by putting the distance constraint, torsion constraints, etc. that we obtained from NMR. This optimization is repeated till all the constraints are satisfied. So till all the constraints are satisfied, ENOE will not be 0. It is only when all the constraints are satisfied, ENOE becomes 0. So the optimization, so from one structure, you get another structure. You further optimize this structure, you further optimize this structure. So the optimization continues in an iterative process till all the constraints are satisfied. So you can see that during the optimization process, initially it was a very random structure. But as you go through the optimization process, it adopts the helical geometry that you expect it to adopt. So this is a single strand of DNA. You can put further constraints about base pair constraints while finding the double-stranded DNA structure. So this kind of iterative process is called the TAND algorithm. So there can also be HSQC. So we have looked at COSY and NOSY mainly, but you can also have HSQC information, which helps you know that what is the nitrogenous base. So here the different arrows are shown. So this hydrogen with this carbon, then with this hydrogen with this carbon, so on. The HSQC data can give you ideas about the correlations between the hydrogens and the carbons. And you can also have 15 correlations between the hydrogen and the nitrogen. So 1H 15N correlations. The good thing is that the chemical shifts you can see here, here the example of adenine is given. They are very unique. N6 of adenine is occurring, N6 is basically this nitrogen. So N6 is occurring in a very distinct, like 82 to 84 ppm. And these two carbons are also very distinct frequency ranges. So you can selectively excite suppose you can selectively excite C2 or selectively excite N6 by using selective pulses, and you can find correlations with that particular nucleus. Similar is the case for all the other nitrogenous bases also. These carbons and nitrogens have very distinct frequencies, and so the heteronuclear spectra, either with 13C or 15N, are also helpful in assigning base pairs. So we looked at the G quadruplex or tetrad briefly. Now we will also look at the different ways in which these can be arranged. So you can have a parallel orientation in which you have 4 DNA strands running parallelly, and the G quadruplex is formed like this, using 1 G from each strand. Then you can have a 3+1 hybrid where 3 strands are parallel in the down position here and 1 is antiparallel in the up. In the antiparallel arrangement, you have 2 possibilities. You have 2 up and 2 down, or you can have alternating up, down, up, down. So 2 kinds of antiparallel possibilities. Then you can have something called a double chain reversal loop. So you have 2 chains here, and the 2 chains are connected by this kind of a loop. So initially, these were 4 separate chains. In this case, you have just 2 chains, and the 2 chains are connected by a loop. You can also have an edge-wise loop. So this is your square, and along the edge square of the tetrad, and along the edge of the square, there is a loop. So it's called an edge-wise loop. Or you can have a diagonal loop. You can also have a single chain. So instead of 4 strands, it is just 1 strand. So suppose we start the strand from here. You come here, there is a loop, edgewise loop connecting here. From here, there is another edgewise loop connecting here. From here, there is a double chain reversal loop connecting here. So the G tetrad in this case is formed by a single strand, and so it's called a monomer. You can also have a dimer where you have 2, like it is formed by 2 strands connected by, each is connected by an edgewise loop. So there can be different orientations of the G quadruplex or tetrad. Now, one thing to remember is that basically you can, when you're looking at the iminoprotons, you will see many more iminoprotons in water. Comparatively, we have already discussed that if protons exchange with D2O, the signal of that proton will disappear. So in water, you will see all the protons, but in D2O, you will only see the non-exchangeable protons. That is why the number of signals in D2O is very much reduced. So here for each G, you will see one iminoproton. We have already discussed that each G has one iminoproton. So suppose you have a structure like this. So you have 12G. So 4 into 3, 12G. So you will see 12 iminoprotons. So the iminoproton region will give an idea about what is the structure of the G quadruplex or tetrad. So suppose you have this kind of orientation. You have two loops that are parallel. The two parallel loops can be on the same side. So you can see the loops in both the cases are going up and then going down. Or you can have two loops on opposite side of the quadruplex. So in one case, the loop is starting from the lowest G tetrad and then goes to the upper G tetrad and again goes down. Or you can have a loop going in the opposite direction from the highest or most upper G tetrad to the lowest and then back. So when you have four G tetrads stacked on top of each other, both these structures can coexist. If both these structures can coexist, so you can see the iminoprotons have been labeled here. Because of the symmetry of this structure, so each small square is a G. This G has an iminoproton number 5. The G parallel to it will also have the iminoproton 5. Just imagine that there is a mirror plane running between here. So 5 and 5 is symmetrical, 8 and 8 is symmetrical. So you will only see, in this case, you will see these signals 2, 3, 4, 5, 8, 9, 10, 11. So 8 signals you will see. And in this case also, there is a certain symmetry, but it is not exactly, in this case, what happens that, again, because of a symmetry, you will see around 8 iminoprotons only. Now, when both these structures coexist, you can see iminoprotons due to both the parallel structure and the antiparallel structure. So the spectra becomes very much complex. You can see here, the G is shown in different colors. And here also, you can see in different colors it is shown. And you can see how complex the structure becomes. One interesting thing is that this kind of G tetrad is generally formed in solutions with potassium or sodium ions. Depending on whether it is a purely G tetrad, it can be a sodium ion solution, or in this case, it is a combination of G and A tetrad, which we'll discuss right now. It can be potassium ion solution. So why these ions are important is that when there is this kind of stacking, the ions actually insert between the tetrads and stabilize the tetrad. So imagine potassium. So depending on the distance between the tetrads, either potassium ion is suitable or sodium ion is suitable for stabilization of the tetrads. Now, here you have, so here all the structures were G4. G4 is forming the G tetrad. Here you have a AG3 structure. So in this structure, something very interesting is seen is that, first of all, the iminoprotons due to G you can see up to 320 Kelvin. So if there was no tetrad formation, if it was just a duplex, you will not be able to see three distinct immunosignals at such high temperatures. They would have disappeared before that. Because you are able to see the immunosignals till such high temperatures, you can predict that a tetrad is forming. So these are the G tetrad that is forming. Now, another interesting thing happens when you look at the NOESY spectra. So in the NOESY spectra, as always, you can do a sequential analysis to find out the nucleotide sequence. But the interesting thing here is that you see peaks of the a NH2 group. So in adenine, there is a NH2 group, and that you can see here. Why this is very interesting is because we have already discussed normally the ANH2, like the amino group is not seen because it exchanges with the solvent. Now, if the amino group is seen here, that means there is something happening that is very unique. So here we have the control case, which is the case which is seen in more than 95% of the time, where amino proton has no peak. You can see the amino proton range, there is no peak. So something very special must be happening in this sequence, that is AG3T, due to which you can see the amino protons. The difference between 1 and 2 is the mixing time. So basically, if we are seeing the amino protons due to some other reason, as you reduce the mixing time, the amino protons should disappear. But because the amino proton peak is persisting even at low and high mixing times, that means the amino proton is not able to exchange with the solvent. That must mean the amino proton is also forming a tetrad. So A, or adenine, is also forming a tetrad. Uh, and this is how the discovery of the A tetrad was done. So A tetrad can form in different ways. So you can see here is the amino group. Basically, it can either pair with this proton of the amino group or with the other proton of the amino group. And both protons of the amino group are thus engaged in hydrogen bonding. And because of that, there is no proton of the amino group that is free to exchange with the solvent. Because the amino protons are no longer exchangeable, you can see the signal of the amino proton. So this is the A tetrad. Now, in this case, basically what happens is that you have both G tetrad and A tetrad forming, and they kind of stack one on top of each other like this. So the red is the G tetrad, and this cyan color is the A tetrad. Because of the two orientations of the A tetrad, there are two possibilities of stacking. Similarly, T tetrad can also form with thymine. So A and G are, you have seen so far. T tetrad has also been observed. Again, there are two possible ways of forming the T tetrad depending on where the hydrogen bond is forming. And if you have a mixture of G and T tetrad, so here what is shown is that you have one DNA sequence, the first DNA sequence, only G tetrad is forming. And in the second DNA sequence, you have both G and T tetrad forming. So how will you know the difference is that in the second case, where there is a mixture of two kinds of tetrads, there is a flattening of the backbone. Flattening of the backbone, you can see here clearly. So this kind of flattening of the backbone is indicative of the formation of more than one kind of tetrad. In this case, it is G and T tetrad. So just by looking at the three-dimensional structure, you can predict that what are the different kinds of tetrads that are forming. And definitely from NMR also you can predict, because if the amino protons appeared in the NMR, that means they are not exchanging with the solvent. And so they must be very much stabilized. So we have seen that if it is just a T bond, one amino proton is always exchanging with the solvent. So in normal duplex DNA formation, amino protons are not observed. So there must be some higher structure more than duplex, due to which the amino proton to exchange, and that higher structure has been observed to be the tetrad that is forming. So we have looked at very basic, we have not gone too much into the details, but we have looked at so many different structures that the DNA can form. And from duplex to triplex to quadruplex. And we have also looked at the basics of NMR. So we have looked at the 1H NMR, the ranges at which the different protons are appearing, the COSY and NOSY spectra, and the NOSY are the most important to find the distance constraints. And using these distance constraints, the optimization algorithm can be done to find the structure of any DNA. So it is from the NMR that these DNA structures are being derived. And we have also briefly looked into how the tetrads were found. So that covers all the things discussed in week 6. I will now briefly discuss some problems of week 5, and then we'll end the class. So in week 5, these are from the week 5 assignments. There was a question that in NOSY experiment, cross peaks are seen between protons, which are, the answer is less than 5 angstrom apart. So NOSY, we already know that intensity of NOSY is directly proportional to 1 by r to the power 6. If the R is 5 angstrom or greater, the intensity becomes too low and it is not observed. So the limiting distance of NOSY is 5 angstrom, which is your option A. And it is also not that we can reduce R indefinitely. R also, so upper limit of R is 5 angstrom. Lower limit of R is also there. It is around 2.4 to 2.5 angstrom. Why? Because of steady clash of the atoms, this is the minimum distance to which atoms can come close to each other, beyond which there is a steric repulsion. This size can vary depending on the size of the atoms, but this is just an average range. So the NOSY distance also has a lower limit, which we anyway saw in the energy algorithm, that the distance constraint has both an upper limit and a lower limit. Then the question is that in 1H 13C experiment, 1H appears along which frequency axis and 13C appears along which frequency axis. So if you remember the experiment, there are two channels in the heteronuclear experiment. Initially, you have a inept sequence where the magnetization is being transferred from 1H to 13C. Initially, you have only the HZ magnetization of 1H. After the inept, when the magnetization is transferred to 13C, you have this kind of a term where, so this refers to the transfer of magnetization from 1H to 13C. Then there is a time period T1 where the magnetization is allowed to evolve in the 13C. And then after that, a reverse inept is carried out where the magnetization goes back to 1H. Now, after the magnetization goes back to 1H, a time period of 2 tau is given. This is basically to bring the magnetization back in phase. So when the magnetization goes back to 1H, you still have this kind of a two intensity term. But it needs to be brought back to the 1 intensity term, which is the in phase magnetization, so that it can be detected. So this time period of 2 tau 2 and the pulse is basically to bring the magnetization back from the antiphase term to the in phase term. And after that, a time T2 is given to collect the FID. So what we are seeing is that in time T1, we are detecting the 13C. You can see the T1 is in the 13C channel and T2 is in the 1H channel. And we know T2 corresponds to frequency 2 and T1 corresponds to frequency 1. So the correct answer is 1H appears along F2, 1H F2 and 13C along F1, 13C F1. So option A. Then you have this question that in 1H 13C HSQC experiment, maximum transfer from 1H to 13C will occur for a value of what? So in the HSQC experiment, basically, this sequence is an inept. So in the inept sequence, the transfer of magnetization is maximum when this tau angle is 1 by 4J. So if we will derive it, the separation between the spins. So basically, J is the angular frequency. J is the difference in frequency, and tau is the time you are allowing for the transfer of magnetization. So this term is essentially 2 pi. J is omega 1 minus omega 2 tau. So this left hand side term is this, and right hand side term is 90, because we have already seen that when it is 90, then this inept has the maximum magnetization transfer. So now if you cancel out pi on both sides, you will find tau is 1 by 4j. That is what this question is asking. So the maximum transfer will occur for 1 by 4j. 1 by 4j is your option D. So the correct answer is your option D. Just give me 2 minutes. Then you have the questions about the 3D NMR. So it is asking that in a 3D experiment, HNCA on a polypeptide chain, correlations are seen between the following atoms of the consecutive residues. So if you remember the HNCA experiment, you start from the, so this is the ith residue, this is the I minus first residue. You start from the amide hydrogen of the ith residue, that is HNI. Magnetization is transferred to the amide nitrogen of the I residue, Ni. From here, the magnetization is transferred to the C alpha of I and the C alpha of I minus 1. So C alpha I, C alpha I minus 1. Then the magnetization again goes back till it reaches the amide hydrogen where it is detected. So the 4 atoms that are involved, shown by the blue circles here, are the amide hydrogen of I, amide nitrogen of I, and the C alpha of I and I minus 1. So the correct answer is option A. Then you have a similar question asking about the HNCO experiment. In the HNCO experiment, you start again. This is the I residue, this is the I minus 1 residue. So you start from the amide hydrogen of the I residue, amide hydrogen of the I residue. Magnetization is transferred to the amide nitrogen of I, or Ni, and then the magnetization is transferred to the carbonyl carbon of I minus 1, or OCI minus 1. So it is called HNCO. CO because it's the carbonyl group. So this is matching with option B, where you have the magnetization starting from the amide hydrogen of I, going to the amide nitrogen of I, and then going to the carbonyl carbon of I minus. So the correct answer is option B. Then you have a 3D HNN experiment. Again, it is asking that correlations are seen between which of the following atoms. Now, in HNN experiment, it is a bit different. So it is a derivative of the HNCA, but it is a bit more complicated. So you start from the amide hydrogen of the I residue, or HNI. It is transferred to the amide nitrogen of I, and this is where you record the T1. So your T1 or F1 is your amide nitrogen of I. Then from here, it is transferred to the C alpha I and I minus 1. This is similar to HNCA, but the difference is that in HNCA, the C alpha is detected. Here, C alpha is not detected. It is just in the pathway of transfer to magnetization. From the C alpha I, this goes to, so there is a NH here, which belongs to the I plus 1 residue. This is I plus 1, this is I minus 1. So from C alpha I, it goes to Ni plus 1. So it goes here. It goes back to Ni. So it comes here. And from C alpha I minus 1, it goes to Ni minus 1 and back to Ni. And from, so now you are detecting in the T2 or F2 axis, you are detecting the 3 nitrogens of 3 consecutive residues, I minus 1, I and I plus 1. And then from these 3 nitrogens, the F3 axis is the corresponding amide hydrogens of I plus 1, I and I minus 1. So in the HNN experiment, you are only detecting nitrogen, 3 consecutive nitrogen, 3 consecutive hydrogen. You are not detecting any carbons. You can see that in the carbon channel, there is no time axis. So all these 3 options get cancelled out because there is carbon, and your correct answer is option A. Then you have another question on the HNN experiment. It is saying that in the HNN experiment, in this plane, what are the signs? Assuming there is no glycine or proline. So what does this mean? The first thing is saying is that F1 is fixed. F1 is Ni, means you are fixing F1. So basically we are looking at this case, which is at particular F1. Then you have the F3, F2 axis. You can see F3, F2 are showing 3 peaks, which are shown in the brackets. They are showing 3 peaks. So what are the sign of these peaks? Now, one thing you can remember as a rule is that the self peak, that is the peak of I with I, will have positive sign. And so we know that from I, you can get I minus 1 and I plus 1. This is what we saw in the previous slide. So this is your self peak, and the other 2 are your cross peaks. The self peak will have positive sign and the cross peaks will have negative sign. So you can just see that that will automatically cancel out your option A and option B. The only option with 2 negative signs is your option C, which is your correct answer. So you can just remember this as a rule, that self peak will have positive sign and cross peak will have negative sign in an HNN experiment. Then these are some, in the last class, we had just gone into the basics of the biological structure. So there are 3 questions based on that. One is just asking that in a DNA double helix, the 2 strands running in opposite directions are held by what base pair? So if nothing is mentioned, we always assume the Watson-Crick base pair, which is the most common. So we know that in Watson-Crick base pairing, adenine binds to thymine by 2 hydrogen bonds and guanine binds to cytosine by 3 hydrogen bonds. So the base pairing is ATGC, which is your option B. Then it is asking that in nucleic acid structure, pseudo-rotation angle refers to what? This we have seen in this class also, that pseudo-rotation angle refers to the geometry of the sugar ring. Because when the pseudo-rotation angle we saw in this class, when it is around 44, it is C2 prime end, around 144, it is C2 prime endo. So the C2 prime is above the plane, C3 prime is below the plane, or the south conformation. And if it is somewhere between 0 to 18 degrees, it is C3 prime endo, that is 3 prime is above the plane, 2 prime is below the plane, or the north conformation. So this pseudo-rotation angle refers to the geometry of the sugar ring. That is your option A. So option A is the correct answer. And finally, you have one question asking that RNA differs from DNA by what? So we know that the one base that is interchangeable between DNA and RNA is that thymine. In DNA, you have thymine, and in RNA, you have uracil. So RNA differs from DNA by the following. So in RNA, thymine is replaced by uracil. And if you look at the structures of thymine and uracil, basically it is this methyl group that is extra in thymine. In RNA, instead of thymine, you have uracil. So where there is AT base pairs, you will have AU base pairs in RNA. So in this case, A is your correct answer. That is, in RNA, thymine is replaced by uracil. So I've covered all the, gone through a summary of all the videos of week 6 and have discussed the assignment problems of week 5. As always, I'll upload both the slides and the video recording, and I'll end the class here.